Hi, welcome to another tutorial. Suppose you have predicted the three-dimensional structure of your protein by using the homology modeling or the threading algorithm, which is implemented in iTaser software. After the prediction, you are completely unaware that how many domains are present in your protein. Or in simple words, you are unaware that what specific function your protein is going to perform. In this case, how we could have an idea about the possible number of the domains and the function of the protein? Is there any way out? Yes, there is a way out. And in this tutorial, we are going to talk about one very important program which is going to help us to solve this problem. This tool is known as InterPro. Now, what this InterPro is going to do? The InterPro provides the functional analysis of the protein by classifying them into the families and predicting the domains and the important sites. To classify the protein in this way, the InterPro uses a predictive model known as signatures. These signatures are provided by the several different databases that make up the InterPro consortium. Now, if you scroll down, then you can see these different databases which are going to make an InterPro consortium. In simple words, your protein sequence is going to be searched for its functional analysis, and then you will have your functional analysis. Now, the use of the InterPro is a super duper easy. You simply need to copy and paste your protein sequence here. Let's copy the protein sequence and let's paste it here in this box. Now simply click on this search button, and when you will click on this search button, then the search will begin. When the search will be done, you will have this sign, and when you will click on this URL, then you will be redirected to your results page. Here you have your results. You can see it here. These are the possible families, domains, and some other features which are predicted by the InterPro. Right now, we are interested in the domain portion. If we will click on this small arrow, then we will have an idea about the possible domains which are present in our protein sequence. If I will bring my cursor here to this pink ribbon, so I will have an idea that the first domain which is present in our protein sequence, this is peptidase S1PA domain, and this spans from the amino acid number 2 to 237. Similarly, if I will bring my cursor here on this portion, so I will have an idea that this part of the protein is going to make another domain which is known as the PDZ. And this domain is present between amino acid number 236 to 327. Similarly, if I will bring my cursor to this ribbon, then I will have an idea that there is another domain which is again the PDZ, and this domain is present between amino acid number 357 to 438. In simple word, in our protein sequence, there is going to be the three domain. The first domain is going to be the peptidase S1 domain. The second domain is going to be the PDZ domain. You can say it a PDZ1. And the third domain is going to be the PDZ, and we can say it a PDZ2. Let's assume I want to have a more idea about the possible function of my peptidase S1 domain or the PDZ domain. In this case, what I will do? In this case, I will simply move to this portion and I will click it here on IPR 009003. When I will click on this one, then I will be redirected to the next page, and on this page I will have a detailed information about the possible roles and the function of the domain in the protein structure. Not only I will have an idea about the possible functions, I will also have an idea about the possible structures of that domain which are available into the protein data bank. You can see it here, there are approximately 3000 various structures related to that domain which is present in the protein data bank. So in a simple word, the InterPro is going to give you a huge amount of information about the possible domains and their function present in the protein structure. Hope so. This information will be very much helpful for you to annotate your predicted three-dimensional structure. And by the way, please let me tell you here that what does it mean by the annotation? The annotation means that the functional characterization of your protein. See you next time.